Hey ANPers, this is Dr. Jones checking in. This is ANP1 Lecture Test 5, Video 20, covering page 21. And the last video that I made actually recorded without the picture of me, so we're going to try that again, and hopefully it records again this time. So, dural folds. So the dura mater is the thickest and most superficial layer of the meninges. The meninges are the term for the three layers that surround both the brain and the spinal cord. They're in the dorsal body cavity, which you may recall when we started the semester in lab and also chapter 1 for lecture. We said there was a dorsal body cavity with three layered protective layers, and in the ventral body cavity we had the double layered protective layers, which were the serous membrane. We learned all about the serous membranes, so now we're in the dorsal body cavity learning about the meninges. The dura are in fact durable, which if we were dissecting sheep brains and when you try to cut through them you'd be like, oh, that's tough. So that's how that works. The dural folds are folds of this tough dura mater that go into the longitudinal fissure and then in between the cerebrum and the cerebellum. And they function to stabilize and support the brain. The brain is fairly soft. It was very, very soft. And of course the skull is hard, so if the brain swirls around too much in there, it can do a lot of damage pretty quickly. We had the picture earlier of all of the blood vessels in the brain, and so once you pull those too hard or smush them too hard, you can get quite a lot of blood inside the cavity, and then of course the skull doesn't expand because of fluid pressure. The brain gets smushed because of fluid pressure, so that's why swelling in the brain is such an issue. So dural folds help to stabilize and support the brain. Dural sinuses are veins in the dural folds. Okay, the first one we're going to talk about is this one right here. That's not a pen. Stop. Go back. Okay, pen. Blue. All right. So this is the falx cerebri. The falx cerebri projects between cerebral hemispheres in the longitudinal fissure. The longitudinal fissure is what we are not calling the brain crack. Thank you very much. The falx cerebri contains the superior and inferior sagittal sinus. The superior sagittal sinus is superior and mid-sagittal. The inferior sagittal sinus is not a lab term. It is inferior and mid-sagittal. So superior and inferior sagittal sinuses, which are mid-sagittal. And they are in the which membrane? The falx cerebri, which separates the cerebral hemispheres. And what is the crack between the two cerebral hemispheres called? That's right, the longitudinal fissure. Number 2 is the tentorium cerebelli. I guess we'll go red now. I'm feeling red. Red. The tentorium cerebelli is this part right here. It separates the cerebrum from the cerebellum. It goes right in that crack there. The tentorium cerebelli separates the cerebrum from the cerebellum. It acts as a tent over the cerebellum. Tentorium cerebelli. Now keep track of your cerebri, which is the falx cerebri was our first one, and the tentorium cerebelli. And our third one is also a cerebelli term, is the falx cerebelli. And it's the smallest one. It's almost like a tiny little sail, like a little bitty sail on a sailboat. And it separates the cerebellar hemispheres. So there's a left and right cerebellar hemisphere, and it's in the tiny cerebellum crack. Thank you very much. Okay, protective function of the cranial meninges. Please read about that. And then let's talk about cerebrospinal fluid. Cerebrospinal fluid, functions of the cerebrospinal fluid. First bullet, it cushions delicate neural structures. 2, it supports the brain. And 3, it transports nutrients, chemical messengers, and wastes. So part of it supporting the brain is that it literally supports the weight of the brain and keeps the bottom of the brain from getting smushed. If you've ever like taken a bath and like just sat in the tub while the water drained, and as the water drains, you get slowly like heavier and heavier and heavier, and gravity like pulls on you more and more and more, because that water added that little bit of buoyancy and helped support your weight. Is the same thing here with the brain. So forming the CSF is the choroid plexus. These bits here and here are choroid plexus. There is choroid plexus in each of the 4 ventricles, and it secretes and monitors the CSF. We know that the ependyma, the layer of ependymal cells that lines the ventricles and central canal, also helps to secrete and monitor the CSF, although the choroid plexus does the majority of it. The entire volume of CSF is replaced every 8 hours, so that is 3 times a day. The lateral and median apertures, now these are not lab terms. Here's the lateral aperture where it goes out the side, and here's the median aperture, which I've already drawn over. So let's get our eraser and back to pen. I'm going hot pink now. Okay, so here is the median aperture, and then the lateral aperture where it goes out the sides. That's an arrow. I can't even draw an arrow. Isn't that terrible? So the 2 lateral and 1 median aperture allow CSF from the 4th ventricle into the subarachnoid space, where it then circulates around the brain and spinal cord. CSF, arachnoid granulations. The arachnoid granulations are on the model, the white dots. And these are areas that absorb CSF into venous circulation. So they take the excess fluid and the wastes and get them out of the CSF and dump them into the veins that are in the dural folds. So CSF is made by interaction between arteries and the choroid plexus. CSF is absorbed by interaction between the veins and the arachnoid granulations. Blood supply to the brain. Cerebrovascular diseases interfere with blood supply to the brain. And a cerebrovascular accident, a CVA, is a stroke. And that's a blood supply is cut off to part of the vein, or to part of the brain. And it can be either like a solid clot that shuts off the blood supply, or it could be like an air bubble that shuts off the blood supply. Air bubbles in the blood supply are bad news. You want gaseous oxygen and carbon dioxide in the bloodstream. Most of it bound to the red blood cells, but we'll talk more about that next semester. What you don't want are bubbles of air. So molecules of oxygen are actually dissolved in the fluid, and like molecules of carbon dioxide are dissolved in the fluid of your soda until you unscrew the top and unpressurize it, and then you've got bubbles. And what we don't want in our bloodstream is bubbles. The bubbles in the smallest capillaries, which are the connections between the arteries and veins, the super tiny ones. The bubbles can block those capillaries, or if they're bigger bubbles, block the bigger arteries. And your red blood cells flow in the fluid portion of the blood. Your red blood cells cannot fly through the air. I'm blinking a third time, so you know I'm serious. Which you cannot see. The red blood cells don't fly through the air. They move in the fluid because they're moving with the fluid pressure, which is forcing the fluid to move through the vessels. When there's a bubble of air, it can get trapped as the vessels get smaller in diameter, and then the red blood cells and the nutrients can't jump across the divide. They can't fly through the air. And so they're trapped, and then downstream, you're not getting fresh oxygen, you're not getting fresh nutrients, and that can cause a stroke, which is cell death. Okay, so on that happy note, that was A&P 1, Lecture Test 5, Video 20, covering page 21. Thank you, as always, for tuning in. I appreciate your patience. The education must go on. Dr. J out.